training in chemistry, structural biology, and biophysics with specialization in the biomolecular NMR spectroscopy. He had previous industrial experience as AstraZeneca UK and Biological E Limited Hyderabad, wherein he was involved in workflows related to innovation and complex generic business. Currently, he leads analytical characterization division of biosimilar business at Biocon Limited. His technical expertise lies in ligand screening, hit validation against different protein classes including membrane proteins, protein kinases, unfolded polypeptides and small peptides, and protein folding phenomena. He has strong interest in drug design and elucidation of structure function mechanism of disease related proteins using biophysical methods. With this short introduction, I request Dr. Vajpai to begin his talk.
Good morning, everyone. I just saw my picture actually. It's probably 10 years back when I was in Vegas traveling. Okay, so first of all, I want to thank Dr. Ashutosh for already bringing my name a couple of times in the slides. You probably will see some of the slides same between us because we collaborate, so we have also copied some of those slides. But anyway, I think I'll try to present my perspective of some of those slides and also will show some additional work which we have done either along with him or separately or from the published material. Right? So, before we go towards the NMR part, I just want to introduce you to the concept of biologics and biosimilar. I'm sure by now you all must have already heard about biosimilars. Is it correct? So biosimilars or biologics are nothing but large molecule drugs which are produced in living organisms, unlike your small counterparts which are chemically synthesized. So because of the nature of differences in these molecules, you will see easier in analytical comparison for small molecule compared to the large molecules. The large molecules are highly complex in nature, and therefore you also essentially need more techniques to identify and characterize them. And these are the challenges which are existing across all biologics, even if they are small, which are peptides, or something like insulins, which are like 6 kilodalton, or even more complex molecular antibodies. So that sort of complexity exists with biologics. Now, what is a biosimilar? Biosimilar is actually a generic version of the innovator biologic drug. So monoclonal antibodies actually came into existence probably 20 years back. And now that they are getting off patented, a lot of companies have emerged and a lot of players have emerged across globe, and they have started developing these biological molecules, macromolecules in the labs. Now, unlike small molecules, the pathways are extremely complicated and not so easy, and therefore you still see very few game players across globe which are working. In India, you probably have heard Biocon and Intas are doing quite well. Dr. Reddy's is there, Lupin are there. So there are only very few players compared to small molecule world, where you see essentially a lot number of companies working on it. So generic development of biosimilar is still, or biologic drug is still very complicated. Okay. So how does it work actually? So I don't know, some of you must have attended other sessions also, but just wanted to show you that the entire life cycle actually looks like this. So if you are working on a biologic drug or a novel drug, the pathway follows this. You develop a molecule, you identify a new molecule, and essentially you have to prove that molecule is consistently coming in the same way, and then you also check for its functional characterization. So there is an activity which is aligned here, but then a huge amount of money you invest in the clinical and non-clinical section, which is immensely reduced and transformed into this, that you have to do a more detailed analytical characterization, and the clinical and non-clinical section becomes small. And it is because of this reason the biosimilars are cheap. So, if you want to say to the regulatory agency across all the big markets, you have to prove that your molecule is as similar as the original molecule. And I am using terms called similar and not same because you cannot produce identical copies. Just like that two humans cannot be identical. Same way, two proteins, because again, they are produced in living organisms, so you cannot have absolute control to produce identical copies. So what you produce is a similar copy of something. But biologic, all proteins, so even the innovator team, for example, they cannot reproduce same identical copy time and again. And we have generated immense amount of data, and we know that even innovators have a wide range, and we, as a biosimilar company, have to match that wide range using all the analytical tools, whatever is possible or feasible for us. Now, okay. So this is a typical product development life cycle, and if you see, you start with drug discovery, essentially you look for a novel molecule, and then of course there is a molecular biology involved which goes into process science, which we are trying to generate large amount of molecule. Then there is the formulation science. Once you have made your molecule, you want to place it into right placebo to actually formulate or inject inside the body. But then we also check for functional assays in the presence of your formulated or excipient composition. You check for PK/PD studies, and then of course clinical, and then so this is entire life cycle. But where is analytical sciences? So actually, analytical sciences touches all domains, and we actually support from the day one of the drug discovery program to the day end. We provide them information, constantly feedback to the process teams to tell them the molecule is coming right along. So it's very, very important. The analytical role here plays an extremely important role for the development of your biosimilar drug. Okay, so how we do that? So essentially, this is a list of techniques which we perform in Biocon or in collaboration with some other external companies, just like Stosh mentioned that we actually also collaborate. So you see, there are immense number of techniques for a characterization of a biosimilar drug. And this is something which we do when we file a dossier from our side. So all these number of techniques are done, including the bioassays. Now, why it is so important? Because it is the structure which covers the function. So if your molecule is not structurally similar to the original molecule, of course your function may get affected. Now, which site of your molecule is perturbed also makes an impact. However, from a biosimilarity perspective, you actually have to establish that your molecule of interest is matching in the fingerprint as similar to the innovator product. Okay. Now, in the afternoon session, I will be talking good about the techniques. But today, actually in this session, I will talk good about NMR. So, are the traditional techniques like CD and FTR good enough? We have to see. So, what does CD and FTR provides you? It provides you information at a global scale. This is what Ashok was telling that you try to match spectral overlap. And if that matches, you say your molecule of interest is actually matching. But what if there are minor differences? Those techniques may or may not be able to be because of the challenges with those techniques and the complexity of these molecules. So you see, in the same low resolution envelope, I can essentially fit this frog to the sea. You see, just if you focus on this, just the movement of one loop on his tongue, he can actually essentially catch the bee. this is the function. If the functional side is perturbed, how can I say that my molecule is actually of the same structure or can behave in the same way? Right? So what we need is to identify right tools which can essentially decipher me the information on this term, whether that is correctly coded or not. Can low resolution techniques provide me that information? May or maybe not. But I will not be confident enough. So I definitely need a tool or a technique which can essentially lead me to this. So what techniques can this be? So I'll just come back to this. So this is a map of the techniques which can provide you high resolution information, also for low resolution. So some of these techniques, CD, FTR, fluorescence, Raman, UCD, they are great techniques because they provide you a quick information at a low concentration, and they are easily available across many labs. So you can essentially get quick information about your product. But is that information sufficient enough? So you actually have to be dependent on some of these techniques. Now X-ray may not be visible all the time. And again, as Ashutosh mentioned, that it provides you a static picture, basically a photograph in your passport. But what you need is a Facebook type of photograph. So whether that is always possible or doable, that is something which you definitely need. So you need NMR, and SDXMS is also extremely important. So I'll touch upon a bit on SDXMS in the later part of my talk. So let's see what are the current challenges. The challenges are, now most techniques when you work with FTIR or fluorescence or UCD requires your buffers to be exchanged. So if my drug product has particular excipient, I buffer exchange them into a buffer which is suitable for that technique, and then I evaluate, compare the innovator product and the biosimilar product. But what if I have lost the real conformation within that excipient? So essentially I am comparing something which is extracted out of the real sample, which may or may not be representing the right conformation. So that challenge is there. Okay. Second challenge is for actually technique like NMR is to evaluate it under the native condition. Native condition, which again, as he has also mentioned, that in NMR what you need is you have to label your sample. Label means isotypically you label 14N to 15N and 12 carbon to 13 carbon. When you label them, your signals enhances and you start seeing excellent spectrum. So that luxury we do not have because we produce products which are non-labeled in nature. So you have to develop techniques which can actually work under native conditions. Third, which is actually very critical, and it's actually very critical for NMR because NMR has a size limit. So if you Google across and you try to see to what extent you have or you can use NMR potentially as a technique and for high resolution, you will notice that NMR actually can suitably work for a protein of 50 to 60 kilodalton. So for going up to 200 kilodalton, you need special tricks to do that. And not all experiments are viable. Only a few set of experiments which you can actually do can actually go up to this. And of course, fourth is, what if I have two spectra? What information I can gather out of it? Whether I say everything is matching, is that sufficient? Or should I develop a numerical tool to actually say, yeah, thoda sa difference hai, but maybe it's okay. So that understanding you need. So, okay, we will go one by one. So I showed you this slide, say that NMR and probably SDX are good techniques to go forward. Okay. So this is a typical pyramid, something which is a figure I showed you in the last page, something similar to that. So you have multiple techniques, as I said, analytical techniques, and if you see at the top of the pyramid are X-ray and NMR because they provide you highest resolution information. Actually, SDXMS can also come to this. Again, there are challenges, pros and cons. it can also be placed in this spectrum. The techniques which are more routine and common are these, but again they are low resolution, as you can see from this angle, right? But still they all provide you complementary information at different levels. For example, polymers and aggregates can be identified nicely by your SEC method or chromatographic method, a night scattering method. You go to the next level, FTR, CDN, always can provide information on structural level. Conformational level, you can get information from these techniques, and essentially X-ray NMR provides you high resolution and detailed information. Please keep asking questions if you have any challenges, doubts. Please.
So nowadays, some of them are it's not possible to crystallize. The people are doing for very effort. So then the people are introducing this technique in the pharmaceutical also.
So cryo-EM, the problem with cryo-EM is still it's not an easy viable. It's extremely expensive, and innovator companies they have started moving towards that direction. But I don't think for a biosimilar industry it is actually a nice tool as of today, unless this becomes a routine, something like NMR and Excel, NMR or maybe say SCXL or something. So at present, cryo-EM may not be the most. It is definitely possible. It is possible, but whether it is possible for routine analysis, maybe one structure you can solve and show that yes, molecule of interest is actually showing the right functional properties.
You know the interactions and everything, either you can visualize it and you get full information for which ratio to solve these things.
Yes. Cryo-EM is excellent tool, and it is in development actually, and probably one day it may become so easy and nice tool as you use NMR.
I just want to comment on this. I think the cost, as you mentioned, is exorbitant, 40 crores to invest in a cryo-EM. And you need to have all the software, everything is available. Technically, it is definitely feasible. Do you require that much amount of high resolution? It's a debate we must have together because of the fact that you've got a lot of functional assays, which also has to be incorporated in the analytical toolkit to assess your function. So the functional assay data can provide you that information. So it's only a nice to have, not a must have. At this moment of time, but later, if things become very cheap or whatever it may be from a cost perspective, then possibly cryo-EM can be considered.
Okay, so now NMR definitely holds an edge because first you can run your sample as such without any change in the environment. So you can take the sample from the syringe, place it in an NMR tube, and add 5% D2. D2 is required because it locks the frequency of the magnet. Without that, you cannot run the experiment. So essentially, you need that. It also is sort of reference for your sample, but 5% dilution will not change anything. So that is okay. Now, because it goes to atomic resolution, you can essentially measure minor changes in the conformation, which is not possible or feasible from the other techniques. One very important point which NMR can also identify is the binding epitope, which Dr. Ashutosh did not mention, but I will show you one slide of that, how we can identify the epitope of your drug product or the other way of product. Okay, so he has already mentioned NMR is actually very insensitive technique. In all other spectroscopes, so actually the reason for it to have such a poor sensitivity is mainly because of the difference in the energy levels. Now, when the energy levels are very little distant apart from each other, your spins are well distributed between the two states. When they are well distributed amongst the two states, even if you apply light, the difference in the population which goes from one state to other is extremely small. And because of such small difference in the population, your signal which comes as an output is also very small. Now in electrons or any electronic spectroscopy like CD, FTI and all those, your levels are too distant apart. So essentially all your population sits here. You apply energy everything goes up, absorbs and comes back, so you require this. So the sensitivity of all those techniques is extremely high. Vanguard slow. Now look at the other nucleus. Okay, this is proton. Proton is hydrogen atom. So essentially proton is most sensitive in NMR spectroscopy. So you see the distance is so small compared to the electronic spectroscopy. But what about other nucleus? Even low. So 13 carbon is almost 4 times lower in terms of this band gap, and 15 is even lower. So essentially, for a 15N spectroscopy, you may need maybe a 1 day or 2 days to generate some decent signal once. While for protons, you can run it in 10 to 15 minutes and you get excellent signals. Nowadays, you can run in 1 minute as well. So with the high magnets and all, you can actually get the same information in 1 minute. And these differences in NMR spectroscopy, Dr. Ashok's already explained that when you place your sample inside the magnet, you have 2 states generated, and these 2 states are also the difference between these 2 states is directly proportional to the strength of the magnet. So if I have a 400 megahertz, my distance will be like this. I have a 600 megahertz like this, 700 like this, 800 like this, 1000 like this. So essentially, you increase the size of your magnetic field strength, you increase the difference, you increase the population difference, and you improve the sensitivity of your signal. So again, it's all correlated, but are those 1000 gigahertz instrument available in India? No. What is the highest speed available in India? It's 800 megahertz. So 800 megahertz probably give you beautiful resolution. You can probably live with it. But again, these are the typical challenges with NMR spectroscopy. Huh? Yeah. So
Also requires
45 crores if you somebody has, then probably purchase a good nice magnet. Okay. So what we can actually done from NMR, right? So you produce a protein. This is a typical thing. You produce. Right? This is done routinely in the labs also. You have a protein. You run an SCS cell. You see a nice band. You say, I can see my protein. But the next part is whether my protein is folded or not folded. So, same protein in an unfolded form has a very narrow spectrum, and protein when it's well folded has a wide spectrum. So, anybody has any solution, any suggestion for why it happens?
Bonds are affected during the unfolded state. So
Something he said, these are basically chemical shifts. So it depends on the chemical environment. So if my protein is unfolded, it will quickly randomly move here and there. So all those interactions are averaged out, and you get a very narrow spectral range for a protein. However, in a folded structure, because they cannot easily go back and forth, your structure remains stacked, and the chemical environment of each of your nucleus is different. So therefore, you get very nice. So this is a take-home message for you. You want to check your protein. Is the easiest way to do it, whether it is well folded or not folded. Right?
Supposedly, like we went from SCS page to NMR. So, according to the current knowledge that I have, after I do SCS page and precise bands, I would think of doing it if I'm only on an enzyme consider. So, I would probably think of doing an activity test to check whether it is the innovator molecule. And if the activity matches, what would be the motivation behind me doing a high order structural analysis after I can match the activity of my protein? Because in the previous lecture or day before, they had also mentioned that subtle differences in the structure do not matter if they do not affect the clinical efficacy of the product.
Understand. So, okay. So your question is well valid. Actually, the thing is, when you have to show a biosimilar product, it's the totality of the evidence which you have to show. Now, functional assays are certainly one good way of showing that your structure properties matches, and structure is very, very similar to that. Of course, this is what I also said in the beginning. where your differences are in the structure, whether is that leading to any difference in the activity is also very important to see. So your question is valid. You may see subtle differences in NMR, but whether that accounts for any difference in the activity need to be checked. So certainly if your activity is matching, you have a way of telling that yes, it is a site of interaction is definitely
way of telling that your
structure
structure
is
is
intact.
comparative structure.
Correct.
I just want to make a comment on your statement. This is a debate we always have in our labs. What happens is that when you do a function assay, you are actually running your sample of a drug in an environment where the excipients are completely absent. So what you have as a drug which gets into the patient is not what you run in a functional assay, because you are diluting the sample to a dose response curve which is not with the excipient. With the excipients will interfere in assay. That's a bigger problem. So what happens is you dilute it with molecule water, you put something else, and bring it to a dose concentration which is amenable for you to run your functional assay, which is not the dose concentration or with the excipient that is being injected into the patient. So actually you are not able to correlate very well. Anything which is functioning well in your in vitro assay doesn't mean that it's going to function well in the clinic. For example, you take a hexameric insulin. The hexameric insulin does not have any binding capacity to the receptor. The insulin which binds to the receptor is the monomer. So what happens is that what you are assessing in a functional assay is a monomer, but what you are injecting to the patient is a hexamer. So this is a challenge. That's where this breaks the gap. So what happens is all these methodologies of higher order structure can be done in the presence of excipients, which retains a fully integrated structure to understand how the molecule is behaving in solution. And that's where this makes a huge difference.
So that is actually very, very important what Ramki said. The concentrations actually play a massive role. So we have been till date, or in fact all biosimilar industries as of today is mainly working with the techniques like CT, FTIR and fluorescence and other low resolution techniques. So high resolution techniques are yet to make a mark in the biosimilar industry. We have actually started doing that. Now slowly we would like to move towards that side, very, in a nice way that we can show all that correlation what you are saying.
Even techniques, CDV probably changed because of the limitations of that technique itself. I get rid of my excipients because of the HD voltage that we talk about.
Yes.
It's like I change the formulation of the protein completely.
Which actually you should not do.
But then that becomes the limitation of that.
Absolutely, that's correct. So you need to look for techniques of CD gives you information, FDR gives you information. But the important point is that you must have in your analytical toolkit methodologies where this kind of dilution of samples do not happen. And you don't have those too many techniques of this kind. One of them could be NMR. For example, let's take the other gentleman was mentioning about cryo-EM. I really do not know because I'm not knowledgeable about cryo-EM. In cryo-EM, I don't know whether you can freeze your drug product along with the excipients because the excipients can trouble you. The reason is that you have got a technology for which the Nobel Prize was given 2 years back. I forget the name, I forget what they call it. Vitrification. Vitrification is very important as a methodology to freeze your samples. At that time, any excipients present in your drug can mess up with your, what you call freezing of your drug substance, which is formed along with excipient, right? So cryo-EM possibly would give you a structure understanding, but is it the correct structure in solution? Very difficult to understand. Only these methods can provide you structure of a protein in solution, which is exactly what's getting into the patient.
Okay, so 1D NMR. So how we assess our product? The easiest way to perform a 1D NMR. 1D NMR is usually, when we talk about 1D NMR, it's mainly a proton weighted NMR spectroscopy. So you see by product, one of the insulin analog is like this, another insulin analog is this. So these insulin analogs are differing from each other only in 1 or 2 amino acids. Apart from that, the whole structure stays constant. But can this technique finds a difference or can elucidate that those differences? So if you carefully look at this spectrum versus this, you will be able to notice one or two changes. But it may not be able to provide you the clear information which amino acids are those. But as a first test, it can still provide you some information. What is interesting is when the same product, same API is formulated, humidity starts in the line broadening. And line broadening tells you that your product may not be in the same state as it was here in this particular case. Which means it might have gone into some sort of oligomeric state, which is actually is a real drug product. So the question again comes back, it again comes back to the same question what she has asked, that in the bioassays, you can check something, whether that is working fine or not, why do we need NMR? So these are the differences what you can see essentially. So this is my API, but this is my formulated product. So you can see immediately a difference. So what is this? So this is high resolution. It is not high resolution because there is a lot of significant overlap, you see. But is this high throughput? Yes, this is high throughput because you can run this in 5 to 10 minutes. So it is fast technique, but doesn't provide you all the detailed information. Okay. So the second one is about the 15N proton correlation spectroscopy, which is HSQC. He was talking about electronuclear single quantum covalence spectroscopy, which is a perfect fingerprint match for different marketed analogs which are present which are currently present in the market. So you see, most of them or all of them are actually on the top of each other, which tells you that structurally all of them are similar. But each of these spectra takes around 96 hours. So yes, this is a high resolution technique, but this is not high technique. It provides you beautiful information, but can we buy this much of time? Maybe not. So you essentially look for a technique which is more high resolution and also high throughput. So again, Toxi and Nosy, Dr. Ashutosh spoke about. You can run on the sample as such, but again, you look at the quality of the spectra, a lot of peaks, very good. This also is a high resolution, but it is also time consuming and it takes around 80 to 20 hours, only 500 megahertz. What Dr. Ashutosh showed was 750 megahertz instrument time, which is again, go from a smaller magnet to higher magnet, you reduce your experimental time. So that is there, but run it on a 500 megahertz, as you see, it runs for 80 to 20 hours. Is it high resolution? Yes, it is still high resolution because at the end it provides you a lot of correlation information, including the information of the disulfide bridges. But this is again not a high throughput. So if you want to develop a biosimilar drug, you also want techniques which can provide you fantastic information, but also can be run in a small time, short time, so that you can actually get good information. Okay, so the strategy, what we were talking about together as team, is about looking at methyl NMR. So again, 6 amino acids, which can actually give you the same information. The advantage of methyl NMR is, see, these are hydrophobic in nature, and your protein folding is highly dependent on hydrophobicity, hydrophobic patches. So your protein not only folds because of hydrogen bonds or electrostatic interaction, but it also folds because of hydrophobic patches. So essentially, all your methyl groups are mostly, they are buried inside your structure and they brings your protein together. So if I see a difference in the methyl NMR spectrum, it means my 3-dimensional structure is actually perturbed, or it's not the same as the yellow structure. So essentially, I have a beautiful fingerprint to even tell whether my protein of interest or the biosimilar product produced by us is same as the yellow product. So I actually have a fantastic tool, and also, as I said, that this can actually provide information also on the binding epitope, which is extremely important. Now, this is one of those tools or technique which can do up to 200 kilodalton. Otherwise, most of the NMR experiment doesn't apply and go to the large molecules. Okay, so this is one good example. Is again a study performed by these guys in say around 2013, using the same tool. So insulin, human insulin, you see, I have shown you a difference between the two sequences. The sequence of human insulin and analog of human insulin, which is the fast insulin, is only between two amino acids which are swapped in this probe. Now, if I run any low resolution technique, because structurally they are so same, I will not be able to find differences between the two. But whether NMR can actually identify that, so we'll just see. So you see, only in this particular region you see a difference between human insulin and this probe. So to elucidate that fact, say two human insulin samples from different companies are run and checked for their methyl spectrum. You see, when the two insulins are taken from different companies, they are banked on the top of each other, which tells me that solution conformation of human insulin in their marketed product is similar. But that is not true, because you see a lot of these peaks have actually moved, which tells you that solution conformation of Lispro insulin is different from human insulin. And that sort of difference in solution in the drug formulated state, no other technique can do. And that is why NMR or solution NMR is extremely important. What Dr. Ashutosh also told you about solid state NMR. So solid state NMR I am not touching here, but also is a beautiful tool to actually identify those differences in the formulated drug product. So is this high resolution? Yes, this is high resolution. Is this high throughput? Yes, this is high throughput. You see, it can be run in 1 hour. Right? It is with the cryoprobe, but the cryoprobe actually improves your sensitivity by a factor of 2 to 3 times, essentially 3 times. So if cryoprobe is not there, you may require slightly longer time, but this can still be done in a couple of hours. So it is extremely valuable. So any protein which is of the order of say up to 10 to 20 kilodalton, I think methyl NMR is a beautiful technique to actually move that. So what is the general perception that molecules actually cannot be you cannot assess molecule with NMR if they are more than 30 kilodalton. So actually it works very fine with less than 30 kilodalton. Then if you want to go to 50-60 kilodalton, you essentially start labeling them. More than that, you actually replace all the non-exchangeable protons with deuterium atom to actually gain in the sensitivity. So you have to play more and more tricks for preparation of your sample, but that luxury we don't have as a biosimilar company. So what we do, we have to think of an alternative technique. So this is what actually Dr. Ashutosh was telling, that you can essentially run this on a map which is 150 kilodalton. Now, what is very interesting is when we run this, we identified few differences between an innovator product and the biosimilar, which is very important because it also tells you that maybe the formulation was not correct, or maybe in the way we have produced this biosimilar product is not correct. So it is very, very important that we have the right tools to actually be able to identify those gaps. Now again, whether those differences what we see here can provide me differences in the bioassays, it can be questioned, because again you dilute the sample and you perform the study. So those are again not so well correlated. But this is the picture actually. So he has already shown this slide. As I said, you may have some slides in common, but you see those excipient peaks can be very well cancelled in your NMR spectrum when you run special, you add a special program to your tool, that is for Sierra, and then you can kill those excipient peaks. See, this is a very neat spectrum. If you want to overlay and you want to evaluate your structure versus the innovator structure, it's a beautiful spectrum. Right? Okay. So as of now, we have understood that NMR is a fantastic tool. Now, if I have an overlay of two spectrum, of course I can qualitatively say my spectra are same. But we also need a tool, some sort of numerical tool to assess my product and also say, okay, this is more similar, this is less similar, this is actually not similar at all. So we need that tool, right? So how we do that? So typically in FTIR, I don't know, some of you must have used it, and if not, then you would be looking for your band, what place your band comes, band 1 and band 2, and wherever they come, we report that value. And simply overlaying the spectra and say everything is qualitatively similar. But then we are specifying a particular frequency and we are saying it is matching. In NMR, actually using a tool, you can actually get the entire picture and then tell whether your molecule is similar or not similar. So MNOVA Bio, together with Brooker, the MNOVA guys have developed a tool called BioHOS. It's an excellent tool. It will take your reference spectrum, which is actually nothing but the spectrum of your innovator product, and peak by peak it will compare with your biosimilar product. And it does this in 3 different ways. One is called principal component analysis. The other one is called ECOS, which is easy comparability of HOS. And the third one is called chemical shift, combined chemical shift difference analysis. So I'll just show you what exactly they do. So in the principal component, how many of you know how to evaluate principal component analysis? Anyone has any info? No? So what it does is actually you have, say, multiple number of spectra and you want to compare one against each other. So I have got, say, 10 spectrum of my reference product, then I have formulated my product and in some buffer A, and I have also formulated, I am trying to screen my excipients. So I have prepared one set of excipients called A, I have made 10 samples out of that. I have prepared in B and C. Now I want to evaluate which one matches more closely to my reference product. So one is I measure all of them, put them on the top of each other and then try to correlate and see visually, okay, it is matching, not matching and all that. But what it does is, it will compare regions by regions and assign your complete spectrum as one dot or 1 point. Now, all my reference product, if they are prepared in a similar way, they will cluster together and they will come at 1 point. Now, A, B, C, if they are made in a similar manner, they will also cluster 1, 2, 3. If all of them are matching very closely to your reference product, they will be very, very close. But if they are not, your preparations will look very different for whichever is not providing the right. So that way you can actually numerically compare between the 2 spectrum whether they are matching or not. So principal component analysis is extremely helpful and is being used when we actually submit our dossiers to regulatory agencies, then we also use one of those as a tool to evaluate our techniques, our data against the reference product. It's very, very important. Okay. The second one is ECOS. So again, I have say recorded spectrum at 37 degrees versus 25 degrees. So proteins, when they are run at higher temperature, they tumble very fast. So their spectral line widths are sharp and peaks also look nice. So the blue ones are sharper than the red ones. The blue one is run at 37 degrees, red one is 25 degrees. So it again matches intensity by intensity and tries to correlate that information whether I have a similar intensity plot or not. So if the 2 spectra are run at 37 degrees, you see a very nice correlation between the intensities of the 2. However, when I have run one at 37 degrees, other at 25 degrees, you immediately see difference in the intensities, and that correlation plot looks not so good. So if I have two different spectral qualities, right away I will be able to measure that there is a difference or not difference. And this is a quantitative measurement. Okay, semi-quantitative measurement of your spectral quality. Third one is combined chemical shift difference. So combined chemical shift difference means I have a peak here, and you see there is a peak which is slightly different. It differs from this particular peak. Now you see there is a difference for this dimension, but there is also a difference in this dimension. So how should we measure? One way is I only look at proton peaks and say everything is fine. But that may not be always okay, because if my peak, instead of moving this side, has moved here. So proton difference is nothing, but carbon difference is there. So you need to avoid combined chemical shift difference. So what we do is we take actually it's like solving a Pythagoras alpha a square plus b square, and then you take the square root, and then you get this diagonal information, and that is what is called combined chemical shift differences. So you do that and you start seeing that differences, that some of those peaks are different, for example, and those peaks also tells you that yes, there is a difference between the two. So if everything is below certain value, now you can say that your protein is actually similar. Right? Okay, so last, I think I have got, I don't know how much time I have. It's done. 5 minutes? Okay, so I'll quickly show you epitope mapping, in which I was talking about. So this work actually we did it when I was working at AstraZeneca. So that time I was working on small molecules. So small molecules, we were trying to identify where exactly it binds in the protein. So we labeled this protein, and I did the complete assignments of this protein, which took around a year or a half to do this. This is a 42 kilodalton protein. When the protein size increases, all your experiment also start becoming more and more purer. But however, this protein behaved slightly okay, so we had gotten a decent spectrum out of it, which I could assign, and we were able to identify each of these peaks corresponding to which amino acid in the sequence. So now, once that information is there, you have almost very detailed information about your protein. Now, when we added a ligand, you see, upon adding ligand, some of these peaks have moved, right? Some of them are on the top of each other still, but some of them have moved. So I again assigned the spectrum for the red one. And when I took the differences in the chemical shift along the protein sequence, I noticed that in some regions you see more differences than the others. And when I mapped the same on the structure, I could identify the binding site of my molecule. And that detailed information again, other techniques cannot provide. Apart from X-ray. X-ray can provide it. And also SDXMS. That also can provide it. So these are the techniques which can again go to such details and provide you that information. What CD can provide you? If there is a structural change. What FTR can provide? Probably again structural change. That they will provide, but so precise information other techniques cannot. In fact, we were able to identify an allosteric change also using a Navaratna chemical shift. You see, all my binding partners are all here, but you still see changes here and here. And this particular site is actually far distant. And when we looked at carefully at the crystal structure, we could see there were hydrogen bonds which were connecting, and perturbation of this molecule or perturbation by this molecule were actually changing hydrogen bond dynamics, and therefore we were able to observe. Okay, so hydrogenation NMR certainly should be used in early phase of development if you want to be sure of your good quality product. I don't know if there is SDXMS I should talk about? Sure. Are you guys tired or can I take 5 minutes more? 5 minutes more is okay? So I'll quickly go through it. So, why I wanted to introduce you to this concept is it's not just NMR alone, while we both work in NMR field, but I think this is also alternative technique which can be beautifully applied across all biosimilar products. So it is getting more and more popular now. A lot of companies have already started showing data based on SDXMS, and recently FDA also have seen that those years come with all these packages, right? So again, as I said, I have moved SDXMS here in this bracket as well, which I did not at that time. So again, it can provide you all that detailed information at the recipe level. And the important point is there is no protein size limit in this case, which is there in NMR. So what you do, how does it work? So you have a protein of interest and you incubate it into because of change in one Dalton, because deuterium is heavier isotope of hydrogen atom. You see some of the protein segments which are more exposed or more flexible, they get deuterated. And which amino acids, which protons get exchanged? They are the labile protons of which are connected to nitrogen atom. So all those exposed amide groups, they get exchanged with deuterium. Longer you leave, more they will get absorbed. So then you see higher hours, you see 4 of those exchanges happen. And when I quench the reaction, and I do that at different time points because I have to check which are more mobile than others. So I do it at multiple time points. I quench this reaction at 0 degrees at a low pH, and then do unfolding followed by digestion, and then perform the MS analysis. So then this MS analysis will provide me information at peptide level. Actually, you can use ETD technique, which is called electron transfer dissociation technique, and you can go to the amino acid level. Sometimes it is visible, sometimes it is not visible. However, if you can go up to the peptide level, that is already a beautiful set of information you have, which you can map onto the structure, and then you would know if there are differences at the peptide level between your product versus the innovator product. So you see, when this has been tested, say for 2 different set of products, and you will certainly notice that between this segment and this segment there were some differences in different formulation profiles. So you want to explore more and check. So essentially, check for which excipient would be the right one to actually reduce those differences in terms of atoms. So that is why it can be used as a tool for also screening your excipients or right formulation conditions. There are some literatures which are given which also show you that even minor differences can be easily taken from this set of data. So what is shown or what is presented usually is a nice butterfly plot, they call it, and each of these line represent different time points of duration. And you see they are actually matching with each other. And if different peptides are checked, all of them show same exchange rates, which tells you that the folding of a protein is similar to the folding of a innovator structure. Again, something similar to NMR, it can also identify the epitope binding. So say I have got a protein which is open here, but the moment I have a drug partner, it goes and bind here, it will restrict those amides which are actually in the binding groove. So I will immediately see a difference between the free form of it versus the epitope versus the bound form, and I will be able to identify the epitope of my drug product. Okay, so again, coming back to the same figure, you would require right tools to identify how the site where the binding happens is different or similar to the inverter. Okay, so NMR and SDXMS both are excellent tools and provide you both low resolution and high resolution information, and both can be used for screening your buffers. And I think it is very, very important that we should use them in the early phase of your pregnancy. These are the people who I definitely want to acknowledge. Our team in Biocon is outstanding, and I did some experiments in Syngene and also with Dr. Ashutosh Kumar. So thank you very much. Please.
So, you prefer using solution NMR with respect to analysis of the in-process DA samples?
Sorry, once again?
For the analysis of the in-process DA samples,
You can actually do that. Very
so
well.
you use it regularly at Biocon.
We haven't done it because we don't have NMR actually in Biocon. We are thinking of it, and you guys should also convince Aarti that we should try it. No, actually, that would be wonderful.
That be actually very relevant. Your question is very relevant.
I have been proposing this as well because that is an excellent tool to know whether your process is coming fine or not. Time and again. So if it is an academic lab, certainly people do it at different stages as well to understand what is the reproducibility of it.
Hi Raman. Thanks for the nice talk. I have one doubt. While we filed the dossier to FDA, we always search for orthogonal techniques data. So what do you think HDXMS data can be a orthogonal data produced in NMR?
Yes, definitely. That would be a wonderful addition if you can perform both. SDXMS as of now we have seen few publications where already people have performed those studies and have submitted them as a part of dossier. But NMR, I don't know whether people have already placed as an information as an orthogonal technique. For insulins, yes. For MABs, I do not know. There are only very little evidences we have seen in literature where they have talked about using the NMR as a tool for MABs characterization.
My second question is in chromatography techniques, we see system suitability data. First, before seeing the data of the samples and method variability. What is like? Do we have? I don't know about any data of NMR. Do we have system suitability data or method variability set in the NMR data sets?
I think that's a very relevant question. So yes, you can actually. So if I want to do the same thing, so we have done it the same thing for insulin actually. What I have done is we take the protein sample and run a 1D NMR and specify some regions in 1D NMR and say that the ratio of this to the reference product should be this much, as well as the spectral should look like this. So that would be my first test to evaluate whether everything is working fine. And then I go for the second test, which is actually time consuming. So why should I waste time when I know the system itself is not working? But there is a publication, nice publication by Kyrie, David Kyrie, his FT guy, who has shown that NMR spectroscopy when run on multiple different magnets over the period of years have shown identical spectrum. So that's the beauty of NMR. You run it anywhere, on any field. Essentially, if your product has not degraded, you will see same spectrum anywhere you run. The difference will happen when your system is not calibrated for temperature, or if your product has degraded. But even degradation of the product, you will be able to notice. That is beautiful NMR. What it will not pick you. So there is a caveat with NMR. If your impurities are of the order of 1 to 2%, I think 2% NMR should pick up, but 1% or so will not get picked up. So you certainly need other orthogonal techniques like chromatography to identify those small impurities. NMR is not capable of doing that. So that is certainly a caveat. So it is an orthogonal tool for giving you information on higher order structures, but it doesn't provide you information on the impurities. Unless you purify them and characterize them to identify where exactly the difference has happened. That is possible.
Without defining a system suitability, you can actually run an internal reference standard every time you run.
Yeah.
So
okay, and use that and generate that data over months possibly, and that will give you a good feel about the reproducibility. Or reproducibility is actually the wrong word, repeatability.
Sir, I have one question. Basically, it is not related to animal. But suppose one of like the insulin is after the storage of 2 or 3 months, there are some minor modifications in the protein. And if you check the binding of insulin to the iron insulin receptors, once there is a slight modification would change in the binding to this particular protein receptor. Then how in industry it will correlate to after changing the structure again in pharmacokinetic studies. They illustrate there is some modifications and effects. So analytical department and the clinical trials, how they correlate.
Do you want to answer this
Oh,
question?
yeah, so
It's a very relevant question.
good question you have raised. So there are two aspects to this. Okay, see, every time when we talk about these functional assays, we are focused only on the efficacy of the drug. You forget that there is something with these biologics which actually Dr. Asutosh and Navartana mentioned is also what is called immunogenicity. Immunogenicity of the drug also can be a problem with respect to drug efficacy. So this is just a comment, okay? Now, you are seeing a difference, but that's the difference you want to see, right? So now what you are doing is better. I hope I understood your question correctly, but what I am trying to understand is, yeah, you are keeping it for stability and then you are seeing a difference in the binding. Right? That's what I'm trying to say, right? That's exactly the information you want, right? So if you keep a drug like that and make it degrade and then you inject into the patient, you first of all won't be allowed to inject into the patients because of the fact that there is something called ethics when you are trying to run a clinical trial, right? As you are aware of it. So what happens is all this data will help you to decide what type of drug and what quality of drug you can inject into the patient. That's the only reason why we do all these things. For example, the HDX-MS study, what we do, we can actually assess stability of our drug using HDX-MS at a very high resolution. More and more sensitive assessment of stability will help us to decide at what age or how much is your shelf life at which you can actually dose your patient. So finally, what are we focusing upon? It's all about patient safety and efficacy, right? Because after some time, many of us sitting in this room become patients. The drug what we are manufacturing. Correct? No, it's going to happen. At some time of our year, somebody is going to go through this, right? So the link is like this. You establish your product quality, then you will be able to correlate to patient safety and efficacy. So from that perspective, the stability data what you're generating will give you the handle, and the clinical team will come back and tell that I won't dose this drug because this drug is a problem. So this will cause problems for the patient.
Thank you very much. We'll keep the questions for the tea break session. I request Nanda to present a token of appreciation to Dr. Navartana.
Thank you.